Hi guys, welcome to Easy Bio. Today we are going to talk about the enzymes. Enzyme, one of the most important chapters in Bio IGCSE O level because it considers as one of the most important basics of biology for the IGCSE syllabus. Before starting the enzymes, we need to know that we have the metabolism, which defined as all chemical reaction going on within a living organism. But metabolism is classified into two different types: anabolic, okay, and this means reaction of building up of large molecules from the small ones. And because we are building up, so we need energy. So our example is building up of glucose to form glycogen, which is stored inside the liver cell and muscle cell. The other type of metabolism is the catabolic reaction, which is defined as breaking down of large molecules into small ones. And because we are breaking down, so it releases energy. Our example, when we break down the glucose by the process which is called respiration into carbon dioxide and water. So we have all these reactions which is important for the living organisms, and they need a substance to speed up this reaction or speed up the rate of this reaction. So we have something called catalysts. What does this word mean? Catalysts mean the substance which speed up the rate of chemical reaction without themselves being changed by the reaction. Okay, this is general. For the whole chemical reaction, we have a catalyst which speed up the reaction itself without being changed, and you have to focus in this point. Because we are living organisms and we have some chemical reactions in our body or in our cells, so this catalyst is called enzymes. So what are the enzymes? Enzymes, protein in nature that function as biological catalyst. So what does it mean? It means they speed up the reaction without being changed. We have two different types of enzymes: intracellular or extracellular. Intracellular means they work inside the cell itself. So they are made inside the cell and work inside the cell. Their action is inside the cell itself. Our example is catalase. What is catalase? Is an enzyme which breaks down the harmful hydrogen peroxide in the liver cell. So we have the catalase as example of intracellular. So it works inside the liver cell itself. The other type of enzyme, extracellular enzymes, which made inside the cell but they work outside, or they released from the cell to perform their function. So what do we mean by extracellular enzyme? The enzymes which made inside the cell and released from the cell to perform their function. Our example is lipase. What's lipase? The enzyme which used to break down the fat into fatty acid and glycerol. Okay, but one of the properties of the enzymes is they are very specific. What do we mean by very specific? So the enzymes for breaking down the fat cannot break down the protein, or the catalase which works in hydrogen peroxide cannot work in glucose, for example. So they are very specific. That's why we have a complex which is called key and lock complex. What do we mean? Here we have the enzyme. As you see in the enzyme, we have a specific structure which is called active site. Because of the presence of active site, the enzymes are very specific, because the shape of active site different from one enzyme to another. Okay, so the enzyme helps the molecules of substrate, this one, to combine together, and they combine with the active site itself. By the time they form the product, and the enzyme will be as it is without any change. So it's called the enzyme substrate complex this one, because the two substrate molecules combine together in the active site, the site of combination between the substrate and the enzyme itself. Okay, so here after that these two products or these two molecules combine together to form the product itself, and the enzyme released and go back to the cell to do its work, to repeat the process with the molecule substrate. That's why they are called key and look, and they are very specific. Okay, what are the factors which affecting the enzymes? We have two factors. We have the temperature and we have the pH. We'll explain in details each other. Temperature. As I told you, the enzymes are protein, and because they are protein, they are affected by the temperature. At the beginning, as you see here, we have the temperature. When temperature increases, the enzyme activities by the time increases, as you see. Why? Because more collisions between the enzyme and substrate. But because it's protein in nature, it has optimum temperature, which at which the enzyme works best. So the product or the production of the product is highest at its peak, at this one. So the peak of the graph. But by increasing more temperature, the enzymes start to be affected. So we have the rate of the activity or the rate of the reaction will be decreased gradually until it reaches zero. What do we mean by zero? It means the enzyme stops completely working. Why? Because the enzyme denatured. What happened? As I told you, we have here in the structure of enzyme, we have a special structure which is called active site, at which the molecules of substrate combine with the enzyme. By increasing temperature more than the optimum temperature, which is specific for each enzyme, so these active sites start to be deformed. So instead of this shape, as you see, it will be different. So when substrate start to combine with this part, which is called active site or deformed active site, it can't be. So the reaction cannot be happened at all. So it stopped working, and we can call it the enzyme denature. In this case, after denaturing the enzyme, because there is deforming of active site, there is no action or activity of the enzymes at all. So this effect of the temperature is permanent effect. So if we are trying, for example, if we have this enzyme X, and this works best at 35 degrees. If we are increasing this temperature to 60, so what will happen? By the time this enzyme will denature, completely stop working. If we are trying to take that temperature back into 35, so even after going back to the 35 degrees, the enzyme it cannot work at all. Why? Because the deformity of the active site or the complete denature, complete stoppage of the whole activity of the enzyme. But if we have the suitable temperature or the optimum temperature of the enzyme 35 and we increase it to 40, so the activity of the enzyme decreases. But if we're going back to 35, it will act again to high temperature, to high rate of activity. Okay, because at 40 there is no denature of the enzyme, so the active site is still as it is without any deformity. I hope it's clear now. The second factor which affecting the enzyme is pH. pH, the same action on the activity of the enzyme on the temperature, but here we have, as we see, we have suitable pH for each enzyme, as the enzymes which works inside our mouth completely different than the enzymes which works in our intestine, so the pH is completely different. So we have, by this is called suitable or optimum pH, the pH at which the enzyme works best, so the rate of the reaction is the highest, the peak. So by increasing the pH, the rate of the activity increases, but increasing more, the rate will be decreased. The rate will be decreased. In case of pH, we have only the denaturing of enzyme at the extreme change of the pH. For example, if enzyme works best at pH 1, so if you change this pH into pH 2, the activity will decrease, but still in acidic solution. So the rate of the reaction decreases, but without denaturing of enzyme. But if we change this pH into pH 14, completely high alkaline, okay, so in this case the enzyme denature, and the action of the pH will be permanent on the enzyme itself, because extreme change of pH on the enzyme leading to denaturing or deformity of the active site. By the way, you have to know that in case of drawing the graph, you have to label the two axes. Because it's very important. Without labeling the axes, the marks will be cut. In case of examples, I'll give you an example from past paper to be clear for drawing the graphs of the temperature or pH, whatever. So I have here an example. We have potato discs which cut, okay, and added in hydrogen peroxide solution. We add thermometer because here we are working in the temperature, and we have here the different variables. I told you before we have two different variables usually in these kind of questions, which is dependent and independent variable. And I told you before the dependent variable which depends on the other one. So we have the first one, independent. As you see, I told you the enzymes affected by two factors, the temperature and pH. And with the change of the pH or the temperature, the activity of the enzymes will be changed. So which one will be the independent one? For sure the temperature or pH. So usually we have the temperature or pH as independent variable. And because the temperature or pH changes, so the rate of the reaction will be changed. In our example, we have our dependent variable here, the volume of oxygen produced at fixed time. And you have to know that at fixed time, because here if we are moving by the suitable temperature, so at the suitable temperature or the optimum temperature or optimum pH, we have the time taken for the reaction is the lesser time taken. So for example, if I have at different temperature 10 degrees, 20 degrees, 30, 40 and 50 degrees. Here, according to the properties of a certain enzyme, we have 30 as suitable or optimum temperature for this enzyme. So if we look at the time taken for the reaction, here for example will be 15 minutes, here will be 10 minutes, here will be 3 minutes, and then 9 minutes and 20 minutes. So from the table itself we can determine which exactly is the optimum temperature if we have this kind of table. The time, this is temperature, and this is the time taken for the reaction. The lesser time taken, it means the suitable temperature or the suitable pH, because here we have the highest rate of the activity of the enzyme because it's suitable temperature or suitable pH. So again, we'll back to this question. So we have here two variables, which one is independent. For sure it will be the temperature, that's why we are using the thermometer, and we have the dependent one, the volume of oxygen produced by the breaking down of hydrogen peroxide into hydrogen and oxygen. So the oxygen produced can be measured, and we have different varieties which should be controlled, like the number of potato discs, size of potato discs, concentration of hydrogen peroxide, and pH. And you should know this kind of questions in paper 6 for enzymes. If the temperature or the experiments on the temperature, so the pH should be constant. If this experiment which is done in the question on the pH, the effect on the pH on the enzyme activity, so the temperature should be constant, because we have two variables. One of them should be constant because the other one is changed. Am I clear? I hope so. So in this kind of questions, I have independent and dependent variable. How can I draw the graph? We'll draw the graph here, the temperature, degrees, okay, and we have the rate of oxygen produced. And please don't forget the labels of and the units of the two graphs. As you see here, this is the independent, so this x-axis, and the dependent in y-axis. Don't forget this, please. So here, if we are trying, we'll see by increasing temperature, increasing the rate of the production of oxygen. So we have this, the shape of the graph, and before drawing the graph, you have to determine which one is independent to be x-axis, which one is dependent to be y-axis. Thanks for watching.